you ready to go? Okay,
Yeah,
well, welcome, folks. This morning, Sam Snodgrass will be presenting on fractionation in the Tripsacinae. One quick reminder is we currently don't have anybody scheduled for next week. So unless somebody would like to strong arm someone or volunteer, I'll remind folks on the Z Evolution Slack that we probably won't have a talk next week, but I would be happy to have a volunteer. And with that, Sam, if you're ready to go, take it away.
great. Well, good morning, folks. Today I'm going to be talking about fractionation in the Tripsacinae. I am currently a PhD candidate in Matthew Hufford Lab at Iowa State University. And this is an ongoing project that I'm really excited to share with you all today. Oop, sorry. There we go. So one of the things that's really unique about angiosperms, when we're thinking about genome size and polyploidy, is that angiosperms have undergone many, many rounds of whole genome duplication events. And so we would expect that their genome sizes would be much larger than what they actually are, given that they've undergone so many duplications. And so in this paper, illustrated it really beautifully, where they actually estimated the ancestral genome size of a lineage before all of those rounds of duplication. And then they estimated what it should be based off of how many rounds of polyploidy it had undergone versus what the actual observed size of the genome size was today. And they found that the current observed genome size was actually very similar to that ancestral genome size. So there's this huge skew towards smaller genomes, much more than we would expect. And that's because polyploidy is cyclical in nature. So in this cartoon example, I've got two diploids here represented by these boxes, which are genes, and the line, the intergenic space. And I've got a reference outgroup that's not going to undergo this whole genome duplication event. And if we compare genes that are related through common ancestry between this reference outgroup and our diploid of interest, we call these orthologs, right? And so after a whole genome duplication event, instead of a 1-to-1 ratio of these orthologs, we get a 2-to-1, where we have two copies of each ortholog to the one copy in the reference outgroup. Each one of these kind of colored boxes representing one copy of that genome of the original diploid, which are now called subgenomes in the polyploid. If that gene order has been conserved in the polyploid relative to that reference outgroup, we can still consider these syntenic orthologs. And when we're talking about the duplicates themselves, we use this special term called homeolog with an extra EO in there, or a paralog. Now, I will tend to use homeolog throughout this talk, but both are seen in the literature. Now, if you give a polyploid enough time, eventually it will start to downsize. And so that just means that it is losing fractions of its genome through these deletion events, such that we're losing these homeologs. So here with the striping, that indicates that that gene is still can be detected, but some of its exons are missing. It's probably not really doing anything. It is on its way to dying and being completely lost. That process being pseudogenization. But in general, we're going to call that fractionation. You can also get lots of chromosomal rearrangements, such as fission and fusion events, tandem arrays, all sorts of things that will break up that synteny to that reference outgroup. Now, this is cyclical, so the ancient polyploid can revert back to a state that is not quite diploid, but diploid-like, in that it looks like a diploid, it acts like a diploid, but it has these footprints of this previous ancient whole genome duplication event. And so in this way, you can have these cycles of polyploidy building on the remnants of previous polyploidy events. And so you might be asking yourself, why are we talking about polyploidy in a Zea evolution talk, right? And that's because the Tripsacinae subtribe, which is the subtribe that includes the genus Tripsacum and Zea, actually originated from a whole genome duplication event. So about 12 million years ago, sorghum diverged from what would eventually become the Tripsacinae. There was these two ancient ancestral species. They're now extinct. There's been a lot of work to figure out what they are, but for here, I'm just calling them ancestor 1 and 2. They hybridized. So one subgenome comes from one ancestor and the other subgenome comes from the second ancestor to create this ancient polyploid. Now, about 12 to 5 million years ago. And then after that polyploidy event, you had the divergence of Tripsacum and Zea, and then the various speciations within Zea. What's notable here is that the sorghum chromosome number is 10, right? That we assume that those ancestral species also had a chromosomal number of 10, so that ancient polyploid should have a haploid chromosome number of 20. Chromosome number in Tripsacum is 18 when you're looking at a diploid Tripsacum species, which indicates that there hasn't been a whole lot of chromosomal rearrangements or fission and fusion events since that ancient polyploidy, right? Those chromosomes, for the most part, remained intact. Whereas Zea, we see massive amounts of chromosomal rearrangements, fissions and fusion events. The genome kind of exploded in a way, but we have now 10 chromosomes. So that is an interesting note to make about these two genera within this subtribe. So fractionation has been previously studied in maize, and in particular, it's been studied between maize lines because that's what we have had the genomic resources to look at. And so here are a couple of the patterns that they have found. So here we have our two subgenomes, our two paralogs, or homologs, if you will, for each line. And if you see a 2-to-1 relationship when you align these genomes to sorghum, that is complete retainment or retention. And if you see that across all of your maize lines, you would say there's complete retention across all of them. Likewise, you can see complete loss, where one of those homologs cannot be detected when you're aligning to that sorghum reference. And so this would indicate, complete fractionation. And these two are the most commonly observed patterns of fractionation that have been talked about in the literature. Though there are other patterns such as partial loss, where if you dig down to the exon level, you see that some of those exons have remained, whereas others have been lost. So you still get some alignment of that second homolog, but it's not a complete set of exons. You can also see in very rare instances differential retention, where one of the homologs is kept in the first maize line and the opposite homolog is kept in the second. Here, the upper maize line has retained the yellow homolog and the lower maize line has retained the blue homolog. And that's pretty rare to observe. So as part of the NAM study, Dr. Woodhouse looked at fractionation within the NAM and aligned all of those NAM founder lines to sorghum and found, so of those 16,000 sorghum orthologs that she aligned to, about half of them were completely lost, where you could only find one of the homologs. But that other half, where you still could find pairs of those homologs, about 4,500 of them, you, for the most part, either see full retainment or non-segregating fractionation, such that that's the partial fractionation, where one of the homologs is retained and the other homolog has some of those exons, but others have been lost, and that pattern of loss is the same across all of these genomes. So that would suggest that loss has occurred before these lines started to diverge from each other, right? But in 494 cases, she saw segregating fractionation, which means that some of those homologs in some lines were retained, whereas in others they were lost, or a different number of exons were lost. And this would indicate that those fractionation events have happened after the divergence of those lines from each other. And so she dug into this a little more and found pretty remarkably that fractionation events are more shared by more closely related lines. So here, if we compare our temperate NAM lines to our temperate NAM lines here in this pairwise comparison, we see that they have much more sharing among them for these fractionation, for the amount of fractionation that they share. And also when you look at, that's true for the tropicals as well. But if you compare temperate and tropical lines for the amount of fractionation they share, it's much less. And this is really a key finding because it suggests that fractionation is an ongoing process and can occur at the timescales of hundreds to thousands of years ago. And this was pretty remarkable because before this, it was really thought that most of the fractionation had been determined and resolved almost immediately after the whole genome duplication event. And absolutely before the divergence of Tripsacum and Zia, that all of this was decided millions of years ago. So this was evidence that it was still happening, which was really, really exciting. But we're also, with the NAM lines, only looking at the divergence times of, a couple hundred to 1,000 years, right? They're very closely related, even though they do include a diverse set of maize germplasm. And so this project is really trying to expand that analysis to the entire subtribe using the genomes that were assembled as part of the Pan-Androbigonia project. And so because of that project, we now have genome assemblies that are chromosome level for almost every taxa within this, every taxon within this subtribe. So we are, for this project, using one of the Tripsacum dactyloides assemblies, one for Nicaraguensis. We got two for Zia diploperennis. Those are pretty heterozygous, and we'll talk about that in just a bit. As well as genomes for all of the subspecies of maize. And so in addition to the existing NAM lines, we have a total of 35 Tripsacinae chromosome level assemblies that we can use for this analysis, which is pretty remarkable. In addition to this, the Pan-Am project also assembled Andropogon virginicus, which is another wild grass species that is about as closely related to the Tripsacinae as sorghum, and can be used as an additional outgroup. So in total, we have 37 genomes with our two outgroups and 35 Tripsacinae genomes. So the goal of this analysis is twofold. The main one that I'm going to be talking about today is trying to understand this tempo of fractionation. When did fractionation occur along that time scale of immediately after that whole genome duplication event through the various divergences of the subtribe down to our current diploid-like descendants. And we're going to be doing this through kind of a parsimony analysis where if a fractionation event is shared between, or identified at least as being shared between two genomes, right, but not the other two, we can place that earlier into the phylogeny versus a fractionation event that is only found within one genome. So to get at this objective, we first have to be able to identify fractionation events. And this really has two parts. The first part is we have to identify deletions. And so we get at this using whole genome alignments using anchor wave. So here in this cartoon, we have a sorghum gene X, with 3 exons represented by these 3 squares, rectangles. And then we have cartoon representations of 3 Tripsacinae genomes. Remember, we're expecting a 2-to-1 relationship. So there are 2 homologs aligning to each one sorghum gene. If a deletion is identified, right, I'm using these lines to indicate that that exon has been lost. And so here we have a couple of deletions identified for exon 2 and exon 3. But we want to make sure that we are comparing homologs from one of the subgenomes all together and homologs from the other subgenome all together, and not mixing and matching across those kind of subgenomes. With more modern polyploids, you can do this by creating gene trees with the known ancestral species or closely related living relatives of the ancestral species that led to the polyploid formation. Because we don't have that for this whole genome duplication event. We have to split these homologs into subgenomes identity by kind of orthology and synteny. And so rather than using some of the other previous subgenome classification that have been created and are still being created for this whole genome duplication event and this lineage, I am instead trying to sidestep all of that and just say, based off of orthology and synteny, these homologs across all of these genomes go together, and so should be in bin 1. And all of these homologs are syntenic and orthologous to each other, so they should go in bin 2 for this one reference, right? And so I'm doing this using a program called GeneSpace. And so we can combine all of this information on terms of deletions and that orthology to get at these deletion calls, where we can then do that sort of parsimony analysis, where we say, okay, for the yellow subgenome, bin 1, those homologs, there's only a deletion identified in exon 2 of species 3. And so that should be placed higher up on the tree as a private deletion. Whereas for homolog 2, which is in blue, we have deletions at exons 2 and 3 shared across species 2 and 3. And so that should be placed kind of deeper into the phylogeny such that it is shared by 2 and 3. So that's the overarching goal of these methods. And one of the kind of tricky parts about this is we want to avoid comparing paralogous homologs across all of these genomes. So let's take that cartoon example I used in the last slide and say that that's the true assignment, that those homologs in bin 1 are truly from the same subgenome, and those homologs in bin 2 are truly from that other subgenome. What could happen is that we accidentally mix up these homologs such that what should be in bin 2 is actually in bin 1, and what is in bin 1 should actually be in bin 2, right? And that would lead us to some erroneous conclusions, such that it would add this fractionation event here. If we're considering all of these homologs as bin 2, it would identify the shared deletion in exon 2, but also add in this private deletion for exon 3. And so if this would mean that we have kind of an increase in the number of private fractionation calls in this example, and would undermine our certainty in the timing of things. And so it's really key that we don't mix up the homologs across these bins in this method, which kind of resulted in this very long and intensive process to identify these fractionation events without using previous classifications of subgenome identity in the Tripsacinae. And we want to make this as robust a pipeline as possible, such that it is as automated as possible, not using any sort of custom scripts, or at least minimizing the amount of custom scripts used. And we want to use existing tools, so we're not building any new tools. And so I want to give a really big shout out to Dr. Woodhouse and Dr. Seathram, who have been really instrumental in helping build this pipeline and think through the framework of all of this. So we start off with our genomes and our gene annotations, and we put them both through GeneSpace to identify those syntenic orthologous blocks, as well as AnchorWave to generate those whole genome alignments. And from there, there is a lot of steps that essentially reduce down to creating those bins 1 and bins 2 for each Tripsacinae genome relative to a sorghum chromosome, and then converting file formats such that the various programs used can interpret the results. From there, we can call deletions using GATK, and from there, filter it down so that we're only looking at genes in the genic or exonic space using the sorghum gene and exon coordinates. And from there, we should be able to get our pan deletion ID matrix, which is the ultimate goal of all of this. While this pipeline does work, I want to throw up a caution sign here, and that I am still working on finalizing and fine-tuning this last step where we're filtering the deletions for gene and exon space, which is something that I will talk about in just a moment. But just a caveat that is the last piece that I'm putting together for this pipeline. But otherwise, everything works. So what I'm going to be showing you today in terms of results is kind of a preliminary proof of concept using just 4 of the Tripsacinae genomes versus sorghum chromosome 10. Now you might ask yourself, why chromosome 10? First of all, it's the shortest, so that helps kind of speed things along. But it also includes this large indel that has been known to cause issues with some of the file conversion steps. I chose these 4 genomes here: Tripsacum dactyloides, Zea diploperennis, parviglumis, and maize, specifically B73, because these genomes span a diverse range of divergence times, which will allow us to test some of these timing analyses, as well as the ability to handle genomes with different chromosome numbers, so that it will be robust in that way. So when we look at the synteny identified by GeneSpace for chromosome 10, we can see that there are these really clear homologs across all of these genomes. So hopefully you can see that there's 2 gold ribbons coming out of this sorghum chromosome 10, matching so that you've got a 2-to-1 match from sorghum to each of these Tripsacinae genomes, and a 1-to-1 match between the Tripsacinae genomes. So to make it a little clearer, we've got 1 copy that matches the entire chromosome to chromosome 13 and in Tripsacum, and 6 in Zea. And the other copy, most of it is contained on chromosome 4 in Tripsacum and chromosome 9 in Zea, with that little chunk at the end there, syntenic to chromosome 10 of Tripsacum and chromosome 5 of Zea. Now, these kind of binnings do match the previous categorization from Schnabel et al. 2011, where here I'm just showing you part of the synteny plots that they created, where you have chromosome 10 for sorghum along the bottom here, and B73 chromosomes along the side. And you can see they identified 1 copy for sorghum chromosome 10, contained within Zea's chromosome 6, and the other copy mostly contained in chromosome 9, with that little tail end in chromosome 5. And so we were able to call deletions across all of these bins, across these genomes, so that we got a total of 82,000 deletions, thereabouts. Now, these deletions are mostly small. The median is about 4 base pairs in length. However, there's a very long tail, which you can see with that mean of about 2,400 base pairs. So a lot of really small deletions with a few really large ones. And this is where I want to throw up some caveats, because this analysis is very, very fresh. We got those deletion calls done over the weekend. And so this is an extremely preliminary analysis, which will undergo substantial revision in the next few weeks. But I still want to show some of these results. So there are some heavy caveats that I want to give before we get into it. First of all, these deletions are relative to the sorghum gene IDs. This does not collapse those gene IDs across isoforms. So there's probably a lot more gene IDs than there are genes. And this does not differentiate between intronic and exonic deletions. This is important because when we're thinking about gene loss and gene death, we're really thinking about the loss of those exons, which are going to have much more functional impacts with their loss, rather than intronic deletions. And so in the future, we really want to focus only on exonic deletions, but here we're including all of them. Finally, fractionation calls here are very, very coarse, such that if there is any deletion within that gene space, that homeolog is considered lost. So here in this cartoon example, you have all of the variants kind of collapsed across all of the genomes here at the top. And then all of these different genomes have different sets of deletion variants within them, but all of them under this categorization would be called lost, even though here they only have intronic deletions, and therefore it should be called retained. And here we probably only have a small loss of this third exon in genome 2, and thus should only be called partially lost, right? So this is very much a coarse collapse of all of these. It also does not account for multiple deletions within a single homeolog or differentiating the different deletions between homeologs across different genomes. It does not account for if that deletion was within a single gene or if it spans multiple genes. And finally, it only considers genotyped deletions. So some of these calls for whether or not the indel was there or not were uncalled, right? So it's unclear if it is a reference allele or an alt allele. And so you get this kind of question mark there. So we tried to ignore those as much as possible throughout this analysis. So with all of that in mind, let's talk about what we found. So the number of deletions per sorghum gene ID, when we're looking at all of them, remember, we've got about 82,000 total deletions. We're not collapsing across isoforms. You get a pretty large number of deletions, about a median of 9 deletions per gene ID, and about 22.8 as the mean. Now, this is not particularly surprising given the number of structural variants known in ZIA, but remember, these are pretty small with a median of about 4 base pairs in length. And so to get at something that might be more meaningful in terms of fractionation, I also filtered those deletions down to deletions that were at least 25% of the length of the gene that they were called in. So this filters those down to about only 9,500 deletions that we're looking at, across about 5,000 gene IDs, again, likely inflated due to isoforms. So we get about a median of 1 deletion and a mean of 1.8 deletions per gene ID with that filter. And that's kind of the filter that I'm going to be using throughout, to make sure that we're only looking at these larger deletions instead of those smaller ones. So we can first start out with just asking ourselves, what is the fractionation status of homologs within a genome, right? Are they considered both retained, one lost and the other retained, or are they both lost? And remember, this is a very coarse analysis where if there is a deletion detected within that homolog, it is considered lost. So what we find is that if a deletion is called in a homolog such that it is considered fractionated for one of those homologs, it's much more common to also see that that other homolog is also lost in this analysis across all of our genomes. My guess is that this is likely due to that coarse fractionation categorization, such that when we make a finer tuned analysis, this number will likely drop. But we do see that evidence of biased fractionation that has been noted within maize previously, where one of the bins is more likely to be retained and the other bin is more likely to be lost. So here we see much more of the pattern where bin 1 is being retained versus where bin 2 is the one being retained. So that seems to hold up. And then finally, this last category where we're talking about both homologs being retained. This is a little misleading in that we are only looking at homologs in this graph that have at least one deletion called within them, or at least fractionated in one of these genomes in one of these bins. So when we look at genes that have no deletion calls or no fractionation across them in any of the genomes, they would not be included here. If we were to include them, I would suspect that they would greatly inflate the number within this category. However, with that caveat in mind, we still see that if there is a homolog that has been called fractionated in at least one of the other genomes, that the Tripsacum genome tends to have retained both homologs relative to the Zia, where that's not as common a case. So this would indicate this is very preliminary, right? And I don't want to make a whole lot of strong conclusions from this, but it would support that perhaps there has been less fractionation in Tripsacum than in the Zias. And this would kind of align with the fact that there's a lot more chromosomal rearrangements, fission and fusion events that might have led to much more gene loss in Zia than there has been in Tripsacum. So something we want to keep our eye on and definitely interrogate more in future analyses. So we can also think about what is the timing of these fractionation events, given this very, very coarse classification scheme. So remember, these homologs are kind of operating independently of each other. So we really have two kind of phylogenies, one for bin 1 homologs and one for bin 2 homologs. And so we can see that the majority of them, if it was called fractionated in one of those genomes for a given bin, it was called fractionated across all of the genomes. So this would indicate a fractionation events that are pretty ancient, which is what we would expect. And we see decreasing numbers as we get kind of further up in the phylogeny and closer to modern day, right? With these decreases. We do see a bump in the private number of fractionation calls. And I would guess that is due in part to there's just more opportunities for something to be called private here versus sister or genus or basal. But I haven't really interrogated if one of the lineages has more private deletions or fractionation events than the others. So something that I'm going to look at in the future. Note that a lot of these are also uncalled. So let's say for say diploperennis, for a given homeolog, it was uncalled for that deletion or fractionation event, whereas it was called across these other three. We can't, because we don't know what the status is for that diploperennis, we can't give a timing for that fractionation event. And so that's what this uncalled bin is. But we also have some paraphyly. And I'll get in and let's dig into that. So paraphyly is where you have a variant or a character that appears in some of your taxa, but not in a way that is a monophyletic clade, right? So in this example, a fractionation event was found in Tripsacum, Diploperennis, and Parviglumis, but not in maize, right? And so you can think about all of the different patterns of which you could have or observe fractionation events across these genomes, and come up with all of these different patterns of possible paraphyly. And so if we look at all the ones that include three taxa versus in terms of these paraphyly patterns, they seem to have more than those patterns that just include two genomes. I'm not really sure why this is. My two hunches are one that this has to do with the fact that we're kind of summarizing all of the deletions and not differentiating between if a homeolog has experienced multiple deletions or has multiple calls within it, that would give rise to paraphyly when in fact it's two separate events. I think it also might have to do with, especially for Diploperennis, it's a very heterogeneous assembly. There might be lots of gaps that could also cause some of this missingness. And I think if those two things are resolved so that it is not the artifacts of how we called these fractionation events, and it is not artifacts of the genome assemblies, then if we still see paraphyly after that, I would suspect that this is incomplete lineage sorting, such that in an ancestral population, that fractionation status had not been fixed, so that there were some members that had the gene and some that had lost the gene, and that kind of continued on through each of the lineages as they were diverging and weren't resolved until later in those lineages, giving these patterns of paraphyly. So that would be my guess if it is not these other artifacts. So I've given you a lot of caveats on these preliminary results. And so I just want to talk about how we're going to improve it. So first of all, we're currently summarizing deletions across the entire gene and using all of these deletions. So the first thing we want to do is exclude those entronic deletions so that we're only looking at the ones that we're really interested in. The second thing we want to do is we want to differentiate fractionation status to the individual exons rather than as a summary across the entire gene. So here, when we do that, we now have different statuses for each exon rather than for the entire gene being lost across all three. And because of this, we now have a finer tuned estimate of the timing of these fractionation events, right? And we get a better idea of them. Finally, we want to incorporate the deletion size so that we get at partial versus complete loss. This could also play a factor into the timing aspect, because perhaps for this exon 3, we'll have to decide how we're going to deal with this, but is this really one deletion across Diploparenus here in this example and maize? Or is this two deletions where there was a genus level fractionation event for the first part of exon 3 and then a private deletion for the rest of that exon? So this kind of gets into the idea of using different deletions or identifying multiple deletions for the same exon to differentiate them. So in this example, it's not really clear, but let's use another example where perhaps we have a paraphily, as currently called, where there's a partial deletion for the Tripsacum genome and a complete deletion for the maize. That, under our current scheme, would result in paraphily, but we can see that these are two different deletion events because they're obviously very different sizes and start stops. And so we can say that these are two private deletion events instead of paraphily. So since I got just a little bit of time, I'll briefly talk about objective 2, which is thinking about fractionation and constraint. The idea being that some genes tend to be fractionated at certain times versus others. So what is fractionated when? And so across several different lineages, they have done these types of analyses where they found that certain gene ontologies tend to fractionate almost immediately to single copy. These are things like essential genes, nuclear plastid interacting genes, DNA repair, and maintenance. On the flip side, there are genes that tend to be retained in duplicate, things like transcription factors and metabolic processes and environmental response genes, right? Things where having duplicates allows for regulatory novelty or neo-functionalization. We can think about this also in a more on more of a spectrum of time since that whole genome duplication event, where you have things that are immediately fractionated to single copy, very, very quick. These are old, ancient fractionation events. And that maybe things that have a really strong constraint to retain as duplicates early on, likely due to dosage balances. Perhaps that constraint starts to relax at some intermediate time frame, and you start to see fractionation of these types of ontologies at an intermediate time since that whole genome duplication event. And then you get into kind of more recent or things that are always retained as duplicates, right? Those things like transcription factors and environmental response genes. These patterns in the literature do appear to be clade specific, but as far as I know, it hasn't been looked at in maize, and especially with the more fine-tuned timing of these deletion events that we will be able to identify from objective 1 and connect to each of these homeologs. I'm hoping that we can investigate these patterns and trends and see if they fit with what is happening in the Tripsacinae. And so with that, I just want to say thank you to the Hufford Lab, all of my collaborators and funding agencies. In particular, I want to shout out Dr. Arun Sitharam and Dr. Margaret Woodhouse, who have been instrumental in both figuring out the framework of this project as well as the bioinformatics part of it. I'd also like to shout out to Elena, who is another member of our lab, who is also working with a lot of the alignment file formatting conversion steps and has been really great to go back and forth with on that. And so with that, I think there are some questions in the chat, but I'm happy to take some more.
Yeah, there are a bunch. I can help you with those, and folks, feel free to raise your hand or put more questions in the chat. The first one in the chat, Sam, is from me on you sort of answered the first part about how often do we see multiple deletions. But I'm also thinking whether it might be worth considering sort of the order of events. And that we expect differential patterns for the first mutation, whether it's a stop codon or a deletion or something that creates a pseudogene versus subsequent deletions and mutations. And might it be worth sort of separating those out?
Yeah, yeah. That is something that we have definitely thought about because what is causing that initial deletion could vary and would also help us understand like how those genes are dying. So yeah, absolutely. That would be something that we could definitely look at and interrogate.
And I guess along those lines, it may be worth looking at other mutations too, because if it may be that the initial mutation is something like a premature stop that creates a pseudogene, and once you have a pseudogene, deletions there are effectively neutral.
Right, right. I guess
Um, Go
would it
ahead.
be
Sorry.
would you be able to see something like a premature stop codon if the gene has been already deleted, right? That would be the tricky part.
Yeah. Right. Right. The Buckler lab asked if the deletion distribution in genes fractionating different from the already fractionated Tripsacinae single copy genes. I think that's referring to you had essentially the SFS, I think, of deletions, right?
Um, so it's more of a it's not an SFS, it's
No, yeah, it was just the density. Sorry, yeah,
yeah, just
but
density of
is it
deletions per gene.
right? Right.
Um, so kind of that difference of partial fractionation versus something that is completely lost. Yeah, so I haven't been able to take a look at that just yet, but that is something that we want to look at so that we can differentiate genes that are in the process of being lost versus things that are already lost. So we haven't quite gotten to that yet, but it is on track to happen.
And then Shujin asks what species are used to form the gene set for gene space. That it looked like 144, but 1,000.
Yeah, yeah. So gene space we use the all of the gene annotations from every genome is used in that gene space. So you identify the synteny between each genome, right, with gene space. And this might be getting into more of the weeds, but essentially we are just using that to form these kind of syntenic blocks where we say the consensus for this given area of the genome seems to be for chromosome 5 in Zia, we are kind of right for here, chromosome 5 in Zia, the consensus is relating to chromosome 10 in Tripsacum.
And so
For
it was just those 5 species that were used for gene space.
um, so we actually ran gene space on all of the genomes, so all 37. And I'm only showing here the kind of results for these 4.
Cool. Okay. Ruben, you had a question?
You can finish the other questions first.
Okay. And then I guess looking at you had your plots that have deletions on the phylogeny showing ILS. And I wonder it might be interesting to look at sort of the expected patterns under ILS. So like that orange pattern where you see 89 losses on bin2, I think under ILS you would expect to see the same number of bin2 losses in the red lineage, the TDZDZB.
Mm-hmm.
And so I wonder if that difference is telling you. I don't know if that represents a problem in the loss in your estimates of loss, or could it be something about selection on one lineage? And I know these are all preliminary, but it might be worth actually, looking at the patterns that you would expect under ILS and asking whether or not where you see deviations from that pattern.
Yeah.
And maybe Kelly, you can jump in. I don't know if this was an answer to my question, but Kelly was
sorry, I can jump in
sure.
on this. This was sort of more just an observation on the paraphyly sort of question there. I mean, if you consider Parviglumis and maize as to just be two, sort of genomes that are segregating within that clade, then it makes the paraphyly issue a little bit more clear, because you actually only, it's on a rotating pivot. And so in a sense, you could think of it as basically the fact that you see the sort of evidence of paraphyly might just be that Dactyloides has had smaller effective population sizes over the past than basically the Zia group.
Okay.
As I don't know, but it might be an interesting thing to think about or test.
Yeah.
And then Shujin mentions that it might be worth splitting out UTRs from other parts of exon, because TEs are more frequently found in UTRs. And you don't necessarily want to be looking at UTR evolution, but rather insertions or deletions in CDSs.
Yes.
And Ruben?
Yeah, I was just wondering, so sorghum goes, genes in sorghum, they are also lost, right? So I was just curious to know, how do you take that into account? Because you may have instances of genes that were lost in sorghum but still present in the CS. So where do you classify those genes, right?
Right. The unsatisfactory answer is we just kind of ignored those, right? So if it's lost in sorghum, we aren't able to necessarily use them here. And that's where having that additional outgroup of Andropogon virginicus, which might still retain that ancestral homolog and has a very strong synteny to sorghum, as well as to the other members of this clade, we could potentially use that to kind of alleviate those problems with just having one, using sorghum as kind of the ancestral state. But we're still working on how to do that. So.
Thanks, Sam.
Yeah.
Let's see, I don't see other questions in the chat. So if there are no other questions for Sam, thank you, Sam. We look forward to seeing the final results of this. This is really cool stuff. And a reminder that please, if somebody would like to talk next week, get in touch with me. But otherwise, we won't have a Z-Evolution talk next week, and we'll be back the following week. All right. Thanks, folks. Have a good day.
Thank you.